Okay, good morning. Again, it's a great pleasure to be here. Thank you for inviting us. So we had a great evening. I'm still recovering last night. So I would first like to thank Spiros for having us here and then being a great host as he has always done that. And then Patri for inviting me to the ETPN meeting. And both Georgina and Alexandra, they both did an excellent job with the meeting. You know, it's very difficult to bring such a meeting together in this excellent fashion. So in this morning, I'm briefly going to give an overview from FDA perspective. You know, a lot of you who are developing these nanomedicine-based products always have concerns about regulatory review. So I thought I would give a brief introduction. And then open up to questions from you, and then join my colleagues, both Simone and Paul, in having a roundtable discussion on the clinical development, preclinical to clinical development of nanomedicines. Again, I'm from the US Food and Drug Administration, which is the regulatory agency in US, where we have many different centers, depending on the product line. It could be drugs, devices, biologics, food, consumer products. So we have 6, 7 different centers, including for tobacco products. That's one of the new centers. And so depending on the product that you come up with, it is reviewed by either a single center or multiple centers. And so I will I guess give the research considerations that you have to really consider when you develop nanomedicine-based products. So FDA's mission is to protect the public health by assuring safe and effective medical products and safe foods for humans and animals. This is our mission, primary mission. In this, within the nano, so I directly go into nano, we don't have any new regulations for nano, meaning the regulations law do not separate nanotechnology products from other products. Because, and then there is no formal definition of nano. But then the decisions, regulatory decisions, always based on, as I mentioned yesterday, 3 fundamental properties of the product quality, the safety of the product, and efficacy of product. Whether it is meant for humans or animals, these are the 3 attributes that we look for when you come up with a submission. Even though we don't have a formal definition of nanotechnology, we have a couple of things that we consider. Usually in US, not usually, but the NNI definition of nanotechnology is any material that is in the size range of 1 to 100 nanometers, where you get unique phenomena that enable novel applications. That's an NNI definition. So we extend the definition in the sense that we consider nano if it is even beyond 100 nanometers up to a micron size. But based on that unique property, there has to be that unique property that is enabling novel applications. So whether it is 1 to 100 or 100 nanometers to a micron size, the end product is to have exhibit properties or phenomena including both physical, chemical, or biological effects that are attributable to the dimension, then it is considered as related to nanotechnology, which is, I guess, one of the major differences from different definitions perspective. So we use the same fundamental questions that we ask about the product quality, the safety, and efficacy. And so how you address them is going to be different. And we understand the complexity that comes from nanomaterial development. So the Code of Federal Regulations, CFR, this requires a full description of the physicochemical characteristics and stability of the drug substance and the drug product. This should include the identity, the strength, quality, purity, potency, bioavailability, the stability, manufacturing process and controls, and analytical procedures. So what does that really mean? That means if you have a small molecule drug, for example, you use techniques like NMR, HPLC, mass spectrometry. to define the product, define the incoming starting material, define the final drug substance and the drug product. In case of nano, you may use additional kind of techniques. For example, if you have a 50 nanometer size particle, you may use, depending on the kind of particle, you may use electron microscopy, you may use a solution phase measurement such as dynamic light scattering, static light scattering. Those are new kind of techniques to define your product. And, but the definition still of the identity of the product, can you specify your product consistently using those techniques? That is a question that you should ask yourself, and as long as you can consistently make the product and then use appropriate tools and techniques and validated methods, then that should be fine for FDA. So again, the validation, this is where standards come into picture, both reference material standards as well as documentary standards. So you have to validate your measurement using an appropriate technique. This is a very daunting challenge for nano. So you have new equipment, new instrumentation coming in, and because of that, there is lack of understanding in some cases as to how to develop a method. It takes a long time, and that is a complexity that comes from developing nano. So for example, chromatography, a traditional chemist or analytical chemists are used to the HPLC, GC, protein purification or characterization can be done by FPLC and size exclusion chromatography, but some of these are now getting into nano. SCC or size exclusion or gel permeation chromatography can be used for certain kind of nanomaterial, whether they're polymers, dendrimers. You can define the polydispersity, purity using some of these techniques. The batch mode measurements using dynamic light scattering, it is very simple. Most people use, and we actually looked at a lot of submissions that are considered nano, and they use dynamic light scattering. It is a very simple technique. You put the vial in, cuvette, and then press a button, it will give you a size. That size polydispersity is very complex. And if there are slight differences in the size, a batch mode measurement does not necessarily detect those changes. It is not sensitive enough. So that's where you have to go through either a fractionation method such as field flow fractionation, size exclusion chromatography, and other kind of methods to look at the polydispersity in the sample. And depending on how you measure, not only you measure and then the instrument spits out a number, but then it depends on whether you are looking at a Z average, the intensity, the volume, or the number. So as you go, a lot of publications you will see a number-based measurements. For example, in dynamic light scattering, which is, from my perspective, useless, because you are essentially erasing the agglomerates that will have a biological impact. If you have larger agglomerates that are not counted, even if you have 5% of those in a number-based measurement. So you have to use appropriate methods to define those products using a more in-depth characterization. So this is, again, a daunting challenge for nano, but it's not to say that this is size measurement is only one property. Beyond size, you have many attributes. Most of the products contain some kind of coating, some kind of surface moiety. You have to see if it is covalently bound or if it is free in solution. In other words, a lot of publications in the literature, they take a nanoparticle, throw in some coating material. And what you have is a cartoon depiction of what you think it is, but that is not sufficient. You have to measure how much of the coating material is there and whether it is free in solution or it is bound to the nanoparticle. The reason that becomes important is the surface characteristics influence biological behavior. In other words, if you take a nanoparticle coated with, let's say, polyethylene glycol or some kind of polymer to passivate the surface, depending on how much polyethylene glycol is on the surface, you can change the pharmacokinetics of the product. You can change the biodistribution. You can change the safety and efficacy of the product. So defining how much of the coating material you have, defining the composition of the coating material, defining the polydispersity, inhomogeneity, heterogeneity of the coating material. These are the things that you as developers should consider, because a batch has a different, it may have a total amount of coating may be the same, but if there is a heterogeneity in the distribution, some of the product is distributed differently because of protein binding, opsonization. Again, depends on route of entry. For example, most of these are IV-infused formulations, and the first biological fluid, they come in contact with is blood. And then blood has a lot of proteins and all kinds of cells. Your immune system responds to anything foreign. So those responses happen because of these, what the immune system sees, and that is the surfaces. And whether it is a small amount of coating or the large amount of coating, the coating density does matter. The size defines your biodistribution, and the coating also defines those biodistributions. So size and coating are the two most important factors when you develop these. But beyond that, the charge does matter. Cationic particles cause hemolysis. They may have cytotoxicity. Shape, we have seen some things with shape beyond the plasmon resonance, for example, for gold nanorods and such. Your biodistribution may be affected by shape, stability of the coating. You may have, what we have seen in some cases is the stability of the coating is critical. You may have the coating when you manufacture it, but then down the road, the coating may come off 6 months down the road. And that has tremendous impact in the safety and efficacy. So again, defining those properties, the quality of the material, safety and efficacy, those are the three things that you have to remember when you come to FDA or any other regulatory agencies. So beyond those nano formulation properties, you have functional properties. Most of them have some kind of therapeutic. In case of cancer, you can localize the drug based on the size. You can change the biodistribution. You can have prolonged half-life in blood based on the size and surfaces with an encapsulated drug that can either accumulate in a tumor tissue using or with the EPR, that is enhanced permeability and retention phenomena that a lot of people use for cancer, or with a targeting ligand. These are the more complex things that are coming. These we call the multifunctional material, where you have a nanoparticle with a targeting ligand and multiple therapeutics. So if you have 2 drugs and 3 drugs that you want to co-localize, and this is a beauty of nanotechnology, where you can co-localize multiple drugs to the site of action and have a synergistic effect to have, to act on the disease. And finally, the imaging agents I will, browse through. So those are the factors that you should consider. But for FDA, the nanomaterial are not new. When the definition, let's say, of nano, 20 years ago, it became sexy, but then we approved nanomaterial-based products for 40 years. And so from that perspective, FDA has a lot of experience looking at these nano submissions. So these are some of the examples. Doxil, you probably know, is a doxorubicin encapsulated liposome. It's a pegylated liposome. The advantage of Doxil compared to the conventional doxorubicin, which is still used, is doxorubicin is cardiotoxic. But then when you encapsulate into a liposome, you get away from cardiotoxicity. You prolong the half-life of the drug. And so this is approved in 1995, more than 20 years ago. And now you have generic forms of Doxil that are in the market. So these are just examples of different kinds of platforms that are inorganic or protein-based particles, polymer nanoparticles, emulsions, lipids, nanocrystals, and micelles. These are all now considered as nanomaterial if you are looking from size definition perspective. So these are all, many of these kinds of material are approved, and we looked at over the 40 years what kind of material or what kind of nano material that FDA is approved, and this is data from that. This is one of my colleagues at Cedar did a lot of work to look at the kind of nanomaterial. So you can see that almost 1/4 of these, more than 300 or so approved products, these are not in clinic, in the sense they are approved for human use. or animal use. And they are 1/4 of them are liposome based. We have nanocrystals, iron polymer complexes. These are all the different percentages, but drug, lipid, micelles, polymer complexes, polymeric nanoparticles, and so on. But if you look at the metallic components, these are really a tiny portion of these submissions are metallic or metal oxide containing. But what we see over the years is that the submissions are increasing at FDA, because it does take a long time, 15, 20 years, for any technology to mature into clinical use. But more importantly, the complexity of the drug is increasing. These are the multi-component systems, not just taking a polymer or some vehicle with a drug encapsulant, but we have seen more multi-component structures. So FDA's questions evolve with the understanding of the technology. So we have more experience, so our questions are also evolving. In coming to devices are classified based on the risk. There are class 1, class 2, or class 3 devices depending on the risk to the public. And again, nano-containing devices have been approved for human use. And these are some of the examples. Now, if you look at these examples, what you see is more metallic kind of particles in the devices area. You have gold, titania, silver, and those compositions are more metallic. And then in many cases, we also have polymeric with various different applications in devices. And FDA issued many guidance documents to help industry. Many of you may know, if you go to the FDA website and search for the guidance documents, FDA issued many, both draft guidance and final guidance documents that are available on the website. You can go to this website. I can make these slides available to you. And the types of submission, again, these are the nanomedicine meeting, are mostly coming through, probably through an IND route if they are new drug application or investigational new drug. So beyond the preclinical work, then you come to FDA, and CDER, Center for Drug Evaluation and Research, reviews these submissions, and those are called INDs before you get into clinical trials. If it is a device, these are called investigational device exemption. So this is an application that you can go through. CDRH is a center that reviews these devices. And then the rest of them are new drug applications. It is approval beyond the clinical trials for sale and marketing. Biological license application is comparable to NDA, but these are specifically for biologics, monoclonal antibodies and protein therapeutics and such. And finally, the abbreviated new drug application, or ANDA, is usually meant for generics. So with that, I will stop here and then open the panel discussion. Again, thank you for your attention. Do you have any questions? Crystal clear. Okay. Thank you. So I will join my colleagues there. The panel discussion. And thank you, Simone Paul, for joining the discussions. I have, maybe we can start with some discussion and then maybe have audience participate. So Simone, I will start with you. The first question I have for you is when you see the nanoparticle submissions, for example, what kind of complexity do you find, and what are the preclinical issues that you kind of consider at EU NCL?
That's a very good question. Thank you. From the EU NCL perspective, I would say we are not that far in terms of applications. The projects usually are really, really early stage in research, and not, for some of them, not even in development. So for for those, let's say, applicants who are going to come to UNCL with the perspective of characterization facility and characterization data, which could, let's say, and should improve their dossier for the application to the regulation bodies, national in Europe. First you go to the national because your first clinical trials are going to be national level, I would say. Those characteristics are going to be key, as it has been described, for several reasons. Let's say the physical, chemical characteristics are going to really modify or impact the biological effect. And since most of our candidates have a drug loaded nanoparticles, you will have also to consider this aspect in the, let's say, road to the clinical development, including the preclinical development, which will be critical in terms of species to be chosen. There is the, let's say, conventional 2 species to be tested and things like that. And considering the pathology you are targeting as well, and most of them are cancer oriented, I would say. You will have to go through from preclinical to the clinical phase, which usually requests some, let's say, key data, the physical, chemical characterization of the nanoparticles, including or not a drug, and the choice of the dose, which is critical, of course. And this is highly impacted from the nanoscale perspective, including the API. So both, let's say, characteristics should be really, really addressed within the characterization of your particles.
Okay, thank you, Simon. One of the challenges we have had is when you take nanoparticles, the first thing is, before you get into humans, the production, you have to produce GMP material. And one of the criteria is sterilization, because it has to be a sterile material. It has to be endotoxin. It has to be within certain levels of endotoxin. And we have seen with many different kinds of metal particles, if you use gamma irradiation, for example, if you take silver and do gamma irradiation, now the structure completely changes. You don't have the silver nanoparticle anymore. Not the case if you have a gold nanoparticle. But certain biologics, obviously, you cannot do steam sterilization. So you have challenges in clinical development versus something in preclinical, let's say, academic research, where these are not as routine. But when you get these into clinical trials, some of these are common things that I think, from my perspective, are issues that you would face both from a nanoparticle perspective as a therapeutic, but as well as a device. Devices also have issues related to sterility. So Paul, let me ask you, do you see from your experience these kinds of issues in, and then what sort of challenges that you see for key enabling technology, multi-kit technologies that you face when you translate things into clinic?
Okay, thanks. So it's one of the biggest challenges, I guess, facing these multi-cat devices. So you take your nanoparticle, you want to make a smart drug delivery system or a controlled release system. Your standard in the medical device industry, the standard is typically either ethylene oxide or steam sterilization or gamma radiation. But any one of those will have an issue. The gamma radiation doesn't like electronics, or electronics doesn't like the gamma radiation. Rather, steam sterilization obviously is an issue with biologics. So I think there's certainly need and room for significant innovation on the sterilization approach alone. The challenge in a way is that many of the new devices will require some kind of a delivery vehicle, which, as you mentioned, needs to pass through a complex manufacturing process where you have traceability and different steps. So it's an unsolved challenge at the moment in terms of being able to sterilize across the different kits, and it comes back to, I guess, when we're thinking about designing and developing new products, we have to design for manufacture, because there's no point in having a working device that hasn't taken into account things like the sterilization, the manufacturability, the design for test at each stage, and so on.
Yeah, I think some of the issues, as you mentioned, seem to be that there is a commonality between even drugs and devices where the sterilization techniques are kind of challenging when you take a multifunctional nanoparticle, where you have a therapeutic, you have a nanoparticle with some coating, and then with a biologic. And then those techniques of sterilization kind of become a challenge. But also the incoming starting material. So in other words, you have to qualify the incoming starting material. At some point, your starting point is some commercial starting material. And then so you qualify given or using some standardized techniques and validated techniques, whether the incoming polymer, incoming coating material, incoming metal nanoparticle, you qualify using validated techniques. And the same is true, I guess, for devices where the incoming material has to be qualified before you assemble them into a device.
And one of the, I guess, the big challenges facing the industry at the moment is the interaction of the combination of the nanoparticles and in particularly the biologics with the substrates that surround it, causing protein aggregation. And so either at the manufacturing stage or in the downstream stage or in the delivery vessels. And so the interaction between the different particles, between the proteins and the formulation, and also between the storage and delivery vessels. And this is particularly becoming an increasing problem as people move from glass vessels to whether it be pre-filled syringes or whatever, to polymeric delivery devices, where again you have new materials and new interactions. And when we almost need to have a real-time sensing to be able to identify the device that has been stored, or the solution that has been stored for 6 months, whether that's now fit for purpose, because typically I think protein aggregation, if it's visible to the eye, then it's deemed not suitable for use anymore, and it's of the order of sub-millimeter aggregations that are deemed to be too much. So we need to be able to mitigate these effects by understanding the dynamic between the particle, the biologic in particular, and the vessels that it's being used in.
Are there any comments or questions from audience at this point? Okay. Oh, go ahead, Ike.
I've heard in the projects a lot about problems with sterilization, and my question is this a topic for research? Could it be a topic for research, or is it a topic that has already had a lot of attention from the research side? Would it be a topic for research to look at sterilization technologies for this kind of situations?
To my knowledge, it's from talking to the industry, it's certainly an unsolved challenge, because up to now, as I mentioned, people have typically been working off of a single type of material or product. In the medical device space, it's precision engineering. In the biologic space, there's a fairly standard process, and people are very conservative in terms of how they've been sterilizing. But as we move into more complex multi-ket type scenarios, it's an unsolved challenge as to how to, and I suppose it's part of the research around advanced manufacturing, how to be able to make such products and have automated processes, and how do you fit this in. But it's certainly, if I talk to the industry, it's an unsolved challenge.
I don't have any more to say, because it's really the key point for us as well, the entry point for UNCL is sterility. If you're not sterile, we cannot move forward with your sample. So it's really key for us, and I think it's key for the applicants to find the best way to sterilize their product. It's not always easy, as Paul just explained.
So to your point, the answer is yes.
Okay, so we needed to put it on the list of priorities for this strategic research agendas.
Absolutely. So there are the two things when sterility, and I'm a synthetic chemist by training, and I don't know anything about sterilization until I got into the biologic world, where endotoxin is a big issue with nanomaterial. And so if you ask the US NCL, and I was there for 10 years, about one single most important issue, Marina Dobrovolskaya, she's one of my colleagues there, she would tell you that endotoxin measurement is a big issue with nanomaterial. What happens is with endotoxin, FDA has requirements based on the potency of the product, how much endotoxin can be present in a product. Let's say for IV infused products. And most of the techniques that are used to measure endotoxin, there is an interference issue with nanomaterial. So your inhibition enhancement controls don't work. In other words, if you spike known amount of endotoxin and then you look at spike recovery, you won't see the spike recovery. So either the endotoxin is binding to the particle, binding to the polymer, that it's not released, so you don't really know how much endotoxin is present. So from a research funding standpoint, if that's a question, I would think the endotoxin and measurements, maybe new methods of measurements, as well as the sterilization techniques may be a good way to encourage people to get in. As we progress a lot of these products into clinical trials, I think that is critical. Okay, so from progressing these into clinical trials, Simone, I have a question for you about phase 0. So this is phase 0 and phase 1 clinical trials. So beyond the preclinical, let's say an SME does a lot of this work and then gets an approval from a regulatory agency. We'll come to the regulatory issues, but what kind of facilities are available or methods in terms of phase 0 and phase 1 clinical trials in European Union?
Very good question. The European environment compared to the US one is not centralized, I would say. It's completely decentralized. So we have national regulations. So as soon as you're going to a clinical trial, you have to face your own country regulation agencies. And it is the case for any clinical trial. Even if you are going for a multi-site clinical trial, you will have to face each country regulation agency. So that's, let's say, kind of a difficulty for the multicentric trials. For the really early phase trial, usually you're a monocentric, so it's at least one national agency, but there are really critical topics or critical points that will be questioned by the regulation agencies. And I think for the nanotechnologies, moreover, regarding the description, the characterization, physical, chemical characterization of your nanoparticles, including the API, meaning what is the role of the API compared to the nano-sized particle you're using as a nanocarrier or anything else, and the interaction, synergistic effect maybe, things like this. So it's going to be very tricky, and they are really going to ask details for that. The biodistribution is going to be hardly question. Where are the particles going? How are they behaving in the body or in the biological environment? And how long do they stay, I would say. The other question is their stability in the biological system, which is key as well. How long does your nanoparticle, or let's say nanosystems, stay as it was injected at the first injection, and what does it becomes? It's as well another question you will face. The pharmacodynamic, including the, let's say, and pharmacokinetic of your compound, but as well as the API and all the let's say, metabolite of your nanoparticle in the API. Regarding the translation from the preclinical to the clinical, the dose is going to be critical as well, because if you're carrying an API, is it the API you will have to dose? Usually yes, because this is the active compound, but when you don't have any API, it's another question. If you have just a nanoparticle, it's really innovative usually, and so you don't have any other solution to compare to. So it's quite tricky, and the definition of the dose to be used as a starting point for the clinical trial is going to be tricky. Even if there are several methods, you will really have to choose the right one and to identify your really good, let's say, API for starting this first-in-man study, which is going to be the first-in-man for most of them.
Thanks, Simon. How about the same for devices? What are the new regulations in European Union? What is the impact on SMEs, Paul?
So this is one of the big concerns, obviously, with the new medical device regulation, because we're trying to find a balance between having safe products for the patient and bringing those products as early as possible to people. And at the moment, there is a big concern with the SMEs that the new regulation will both delay the introduction of products, but also impose quite a significant cost burden in terms of the extra details required. This is particularly the case for class 2 and class 3 devices. And I think there are a number of issues. One, in terms of the classification, my understanding is that many of the devices will end up as a higher classification, in this case class B to D, whereas, for example, in vitro, previously most of the devices would have been in the lower classes. So that's one change. So now you're dealing with a higher requirement in terms of regulatory approval. The clinical evidence requirement will be much more significant, and it needs to be updated throughout the life cycle of the product. So again, for a small SME, bringing a product to market would both have time and cost implications to do this to get this clinical evidence. There's a requirement for traceability, so no longer will it be acceptable to have a batch level traceability, but you need individual device traceability. And this is already standard practice, for example, in some of the class 3 devices like pacemakers and so on. But as we move forward to surgical instruments and so on, it becomes much more. There are significant impacts in terms of manufacturing costs and so on to be able to track each device, and if there's a defect, to be able to go back and look at which component was the defective component. If you're following the story with the Galaxy Note at the moment, you could understand why you would exactly want to pinpoint where the issues arise. And in many cases, companies will have multiple suppliers, and so you would want to be able to identify, based on the quality controls of those sub-modules or components, where the issues were. And that's another part of it, is the requirements to monitor and validate the supply chain will be much more extensive with the new regulation. And again, there are issues around companion diagnostics and so on. They would be the main impacts for an SME. So from a practical perspective, it's about the cost and the time.
Thank you, Paul. Robert?
Just a quick comment. We have 5 more minutes.
Yes, sure. Yes.
Okay. Just one question to Simon, and a comment on what Paul was saying on the devices. Simon, you were saying that for a multi-center trial you have to face all the countries. I believe in the relatively recent clinical trial regulation, it was included that you have one entry point if you had a multi-center trial. I believe that is possible. You can choose the reporting country, so perhaps we should check it. My computer is currently dead, so otherwise I would have checked it right away. But please take a look at that. And the same kind of process has been included for the devices regulation also. The clinical trial regulation purely for pharmaceuticals, but the same kind of procedures have been taken over in the new MDR. And I think if you're talking about indeed there are a number of provisions that are strengthening the requirements for clinical evaluation. And I think an important point, Paul, that you have not mentioned yet, is that for class high-risk devices, a number of them, there are some exemptions, there is an extra requirement that it has to be reviewed by expert panels. So there will be an extra layer. Expert panels will, well, they have not been installed yet, of course. That needs to be done in the upcoming years. But there will be mostly clinicians probably in these panels. So especially for the high-risk implants, there will be some extra issues. And if you look at the nano, there are specific provisions for nano also with regard to the risk classification. In the original proposal by the Commission, it was simply stated that all devices containing nanomaterials would become class 3, that is the highest risk, where a nanomaterial was defined according to the definition in the recommendation of 2011. And so that was quite difficult to enforce. And there have been long negotiations with the European Parliament and the Council, and it has now been changed into a classification depending on the potential for release of the device of free nanomaterials, and then you are in different classes. So for anyone who has devices with containing nanomaterials, it's important to take a look at the new texts for the MDR.
Thank you, Robert. Any comments? Any other comments?
I was just going to say that not specifically in the nanospace, but as an SME, we have noticed that the European regulation framework, and colleague over there gave a good explanation of some of the changes, but I think a lot of those haven't yet come into force. But we still notice already a change in the interaction between us and the notified bodies, and it's getting tougher. There's no doubt it's getting tougher, and I mean, a classic example is we did one product. We started at the same time with the US and over here, and we got approval through the FDA quicker than we did in Europe. And I think that a few years ago that would have been quite rare. I think now it's probably going to be more common. And we're having to, I think our experience is we're having to feel our way a little bit more than perhaps we thought we would previously. So I think the device world is getting more complicated over here.
Go ahead, Panos. Maybe we have 2 more minutes left. Yeah, 1 minute.
Anil, a very short question. Do you have any idea about the borderline nano devices? I mean, nanoparticles that they have switch on, switch off mode of action, and they cannot be characterized by the primary mode of action being pharmaceutical or immunological or metabolic. Do you have any, let's say, comment to say from FDA perspective, if you are regulating this, do you have regulated any nano device, a nanoparticle that is not a pharmaceutical, is a device?
So if your question is, did we approve a nanoparticle as a device? Yes, we have some examples of that. So when submissions come in, and if some of them are cut and dry, we know it's a drug, and in some cases we know they're devices, there is no question. But there are gray areas whether it is a drug or a device, and then we have an office that looks at these submissions and then makes a determination as to, based on the primary mode of action, whether it should be considered as a device or a drug. And in some cases, if there is a device containing drug, both CDRH, that's a Center for Devices, as well as Center for Drugs, they come together, multiple centers within FDA, and then review the submission. So yeah, we have examples of nanoparticles that went through as a device rather than a drug route. Okay, I think we ran out of time here. Again, I thank my panel members, Paul and Simon, for taking time, and then thank you for your active participation.